Hi guys, and welcome back to my channel. In this video, I will be going over the third line of defense our body has against pathogens. And I'm not recording a video on the first and second lines of defense. We did go over those in class. So this will be specifically focused on the specific immune system. This is the portion of our body's defenses that can specifically seek out particular pathogens that can invade our body and then destroy them. So this group of cells that we're going to talk about, they have this ability to kind of do this search and destroy type of defense. That the first and second lines of defense, the physical barriers and the internal barriers, just don't have that kind of specificity that this third line of defense does. So these are the PowerPoint slides that I have uploaded to Canvas, so you guys can download those if you wish to. And since we will not be having an additional lecture on this, this is the lecture. So if you typically take notes in lecture, this could be a good time to do that. Or as you know, this video will be available to you for a long period of time, so you can just come back to it if you want to hear it again. Okay, cool. So let's get started. So on this slide right here, this is kind of the intro to this section slide. So the first thing we have on here is that our immune system is a population of cells. That's what this says here. Let me get my drawing tool going. Okay, so our immune system is a large population of cells. Okay, and so these cells are found in all the organs of our body, but are very highly concentrated in specifically the organs of our lymphatic system that we've talked about previously. And these cells have a really cool recognition ability. So this is what we're going to be discussing in the section. Okay. So these two characteristics here are going to distinguish immunity from non-specific resistance. Immunity is this specific search and destroy feature of the cells we're talking about, and non-specific resistance is the other mechanisms we talked about previously, like fever, natural killer cells, physical barriers. Those are the mechanisms of non-specific resistance, and today we're going to focus on specific resistance. So our immunity, or the cells that participate in immunity, are cells that have specificity. They have immunity directed against a particular pathogen. This is that search part of this mechanism, where the cells actually search for specific pathogens in our body. Okay, so they have specificity. They can specifically recognize certain cells. And these immune cells that we're going to talk about also have memory. They can also remember a pathogen so that when we're re-exposed to it again, the body can react so quickly that we don't even have any noticeable illness. So this is like, when we get the chicken pox the first time when we're a child, we get really sick. But then our cells store a memory of this pathogen. And some of the cells are specific memory cells that live in our body for a very long time and can, if we ever get the chicken pox again, they can initiate an immune response so fast that we don't even know that that pathogen has invaded our body a second time. So we don't get any noticeable illness. Okay, so going on to the next slide here. So on this next slide, we have here an outline that kind of shows us the different types of immunity that our body has, or that our body can develop. And so let's see. Okay, so on this side over here, let me get my pen again. Okay, on this side over here, we have our innate or non-specific immunity. This is the first and second lines of defense that we previously talked about. This is genetically determined. This doesn't require any prior exposure to pathogens to develop. So we are born with this and we have it from the time we're born. Now, this side over here, this is our adaptive immunity. This is what we acquire. We can also call this our acquired immunity. This is what we acquire throughout our lives. You know how little babies don't have a very strong immune system, and one of the ways they get their immune protection is from their mother. So little babies, they only have this kind of immunity over here, this innate immunity. Innate means we're born with it. It's like intrinsically within us. So this we have from birth, and this one over here, the adaptive immunity, we acquire. Now, there's two types here of adaptive immunity. We have active immunity and passive immunity. The active immunity is immunity that develops because we have been exposed to some foreign antigen, some foreign marker on a foreign cell or a foreign protein even. So this is something that our cells are actually working to give us immunity from that foreign molecule, okay? It could be a pathogen or even just a protein from a pathogen. Passive immunity is not involving any real work by our cells. Passive immunity is produced when antibodies, these little protective protein molecules, are transferred to our own body from another source. So in this case, in passive immunity, our cells aren't really doing much to protect us. We've just acquired some antibodies. In our own body, we've acquired some antibodies that are going to passively protect us. Okay, so one of the types of passive immunity is down here. It's naturally occurring passive immunity. Okay, this is here conferred by transfer maternal antibodies across the placenta or in the breast milk. So in infants, they don't have their own active immune system yet. So we don't have any active immunity over here going on inside infants. Instead, their immunity is passive. In the breast milk from their mother, they have antibodies that come into their body and actually will attack pathogens if they catch some kind of bug. Okay, so if they catch some kind of pathogen, those antibodies can protect them passively. Their own cells aren't doing anything to protect them at this point. They're just having this passive immunity from the antibodies. Another type of passive immunity over here is artificial. Okay, so this would be artificially induced or artificial passive immunity. This is, we see an example of this when we medically will administer antibodies to a person who has acquired some pathogen that could be really, really deadly, and we want to give them some very early protection against that pathogen. So we do this in if a person gets rabies, or if we suspect a person may get rabies. Because rabies is so deadly and could kill them so easily, we want to start treating them immediately for that. We don't want to wait until their own cells, which would be inactive immunity, this is involving cells, we don't want to wait until their own cells are attacking the pathogen, because they will already be too sick at that point. So instead, we're going to do this artificial passive immunity where we give them some antibodies. We're going to administer some antibodies to them. And this is going to begin protecting them right away from rabies. Okay, so their cells aren't involved at that point. So these are types of passive immunity, not involving cells. They're involving antibodies acquired from some other source. In active immunity, in this case, these are mechanisms in which our cells are actually involved in. Active immunity is immunity that is acquired because our cells encounter a pathogen or part of the pathogen And those cells actually react to that pathogen. So they recognize those pathogenic molecules, and then the cells develop an immune response. So in the body now, we have cells stored that remember that pathogen. So active immunity actually involves memory. The passive immunity does not. Okay, so active immunity involves memory. So these cells, they're exposed, they develop an immune response, and then they remember the pathogen. So here's one example where we have naturally acquired immunity. And the naturally acquired immunity occurs naturally. So this is if we just, catch a pathogen. Let's say we catch a cold virus. Okay, a cold virus enters our body, and so we're exposed. Okay, we're exposed to this cold virus. It comes into our body, and our cells begin to attack it and any infected cells to destroy the pathogen and eliminate it from our body. So our cells develop an immune response. We get sick, we experience some symptoms, and then we have a memory of that pathogen for the future. So we've naturally acquired this immunity. We also here have artificially acquired active immunity that develops if we have some artificial administration of that pathogen or part of that pathogen. So this is what occurs when we give someone a vaccine, because vaccines are essentially parts of viruses or weakened viruses that we inject into the body. And so they can't really infect us because the whole virus isn't there, or the virus is severely weakened. So we're actually not going to be infected. We wouldn't get completely sick like we would if we naturally encountered this virus, but our body will have an immune response, a weak immune response. So our cells will react to the virus and they will remember the virus, just like we have in this other case over here. So in both of these cases, we develop a memory of the virus so we can protect ourselves in the future. The passive immunity doesn't have any memory, so with passive immunity, these mechanisms do not allow us to protect ourselves from the virus if we ever encounter it again. Okay, let's go on to the next slide. All right, so we're going to talk about, with our acquired or our adaptive immunity, we're going to talk about these two branches of it here. So if we have here our adaptive or our acquired immunity, okay, we're going to start with something called antigen presentation that we're going to look at shortly. But basically, once our body recognizes that there's some foreign antigens in it, we're going to trigger some defenses and we're going to get an immune response. And this immune response has two parts. Okay, we have the cell-mediated part of our immune response, and we have the antibody-mediated part of our immune response. The cell-mediated immune response involves T cells. The antibody-mediated immune response involves B cells. Okay, so I would just memorize this right off the bat. Cell-mediated immunity is T cells. Antibody-mediated immunity is B cells. We also call antibody-mediated immunity humoral immunity. And let's see here. Okay, so let's start with the cell-mediated immunity. This involves the T cells. The T cells actually will directly attack the foreign cells and any infected host cells that we have. So this involves the T cells actually directly removing and destroying. So here we have destruction of the pathogen. Okay, so we're actually removing and destroying the pathogen. The antibody-mediated immunity with the B cells, this uses antibodies to attack the pathogen, and antibodies on their own cannot destroy anything. Okay, all they can do is bind to the pathogen and kind of target it for destruction. They're kind of marking the pathogen, saying this needs to be removed from the body, and then T cells or macrophage will come along and destroy those pathogenic cells. So antibodies can't actually destroy anything. They can just bind to infected cells or pathogenic cells and kind of mark them for destruction. Okay, but both of these branches of this acquired immunity will result in the destruction of the antigens, okay? And so both of these parts of the immunity also will recognize the pathogen, react to the pathogen, and then finally remember the pathogen. So these are kind of the 3 R's of our specific immune response, that the cells recognize, react, and remember the pathogen. Okay. So this is going to begin once our cells will recognize specific foreign antigens. Okay, so these cells have to be able to recognize foreign antigens. And I know we've talked about antigens before because we talked about the A antigen, the B antigen, and the Rh factor in blood typing. But I just want to go over really briefly what an antigen is in case we forgot. So an antigen is some little molecular piece of well, in this case, of some foreign particle, okay? It can be something in our own body antigens, but we don't want to focus on our own antigens right now. We want to focus on foreign things because we're talking about foreign pathogens here. So these antigens are actually small molecular pieces. It could be a little carbohydrate from a bacterial cell, okay? Or it could be a little protein from a viral particle that we're recognizing. That's going to be some little molecular piece of this foreign entity. Okay, let's go on. All right, so let's start with here, talking about our T cells, our T lymphocytes first. Okay, and we want to kind of look at how they develop and how this is going to make them very well suited for the job they have in our body. So these T cells, they're actually, we say that they're born, okay? They first develop in the red bone marrow. And I know we've talked about T cells developing in the thymus, and that's true, but they first start out their development in the red bone marrow. They are actually descendants of the pluripotent stem cells. Remember, we talked about these when we talked about red blood cells. And those pluripotent stem cells live in the bone marrow. So the T lymphocytes are born there. And then they are released into our bloodstream as pre-T cells, a kind of stem cell. And they're released into the bloodstream as some precursor to T cells, stem cells, that travel through the thymus. Okay, and first they reside in the thymic cortex. Let me get my pen. Okay, so first they reside in the thymic cortex. And so this is the superficial portion of the thymus. It's kind of the outer portion of the thymus. Okay, so they're living in the thymus, and here they will go through a process in which they become what we call immunocompetent, this word right here. Okay, they're going to become immunocompetent. And when a cell becomes immunocompetent, this means it's developing receptors for surface antigens. So here I'm just going to draw us some different kinds of T cells. All right, so these are all going to be T cells. Okay, and so this happens throughout our lives, but especially very early in development, like when we're an infant, okay, when our immune system is developing really strongly. Okay, so these are all little new T cells. They're living in the thymic cortex, and they're going to become immunocompetent. And immunocompetent means they're going to develop these little surface receptors for foreign antigens. So all the different T cells are going to develop different cell surface receptors. So this T cell might develop receptors for one specific foreign antigen. So maybe this receptor has one molecular shape, okay? And this T cell is going to develop receptors for a different surface antigen, foreign surface antigen. All right, and here we have this little guy. And this T cell is going to develop different cell surface receptors. Okay, I hope this makes sense. So all of our T cells are developing a different specificity for some foreign markers. These T cells, we're basically creating so many T cells that we essentially have a library of T cells in our body, like a T cell that could recognize any potential foreign antigen we could ever come across. Okay, so we're talking millions of T cells, or we have many, many different kinds of T cells, and all of them have a different type of cell surface receptor. Let's say maybe this one has like this type of shape, okay? And so we're potentially creating a different cell surface receptor on every T cell, theoretically here, that could recognize any type of pathogen we could ever come across. I know this seems really wild, but basically we have a library of T cells. Okay, so these T cells, they're stimulated to develop these cell surface receptors by these cells called reticular epithelial cells, or RE cells. Okay, these reticular epithelial cells, they live in the thymic cortex, and they're secreting these hormones called thymosins, and the hormones are stimulating the T cells to develop these receptors. You guys are going to need to remember thymosins for the last chapter of the semester, so this would be a good thing to memorize now. Okay, so the thymosins are telling the T cells develop all these different cell surface receptors. These reticular epithelial cells, once the T cells are created, the reticular epithelial cells are going to test the T cells. Okay, they're going to test them, and they're going to do a type of test we call negative selection. This is where they will present the T cell with some type of antigen. Okay, so let's just draw a little picture here. So if this is our T cell, okay, and here's its cell surface receptors. So it's newly developed. And here's the reticular epithelial cell. Reticular cells, the word reticular is meaning that it has like a branched sort of star-like shape, but we're drawing it as a circle. Okay, so the reticular epithelial cell is going to present this T cell with some surface marker, okay, some type of protein we have here on the surface of our reticular epithelial cell. So it's actually binding to the T cell. And the reticular epithelial cells are presenting the T cells with our own antigens, so our own host proteins. Okay, so the reticular epithelial cells, they're basically saying to the T cell, do you recognize this host protein? Do you recognize this own protein that belongs in our body? And the T cell is supposed to recognize our own host proteins. Okay, so it should be responsive. Any unresponsive T cells are eliminated. They're going to be destroyed. Okay. The T cell should also, not only should it recognize our own host proteins, but it should not react to them. Okay, it should not try to kill the epithelial cell. It should just recognize our own host proteins, but not destroy them. So any self-reactive T cells, this means that they are trying to attack our own cells. Let's say the T cell thinks that our own host protein right here is foreign. The T cell will try to attack the reticular epithelial cell. If the T cell does that, the reticular epithelial cell will kill the T cell. Okay, so we want T cells that recognize our own host cells but don't kill them. And so we test for this with this negative selection test. This is performed by the reticular epithelial cells. Once the T cells pass this test, then they move to the thymic medulla. This is the deep part of the thymus. And here they're going to multiply and form clones. So they're basically producing many, many copies of themselves. And this is going to basically pack our body full of T cells, full of these T lymphocytes, so that if we encounter a pathogen, the T lymphocytes are there to begin an immune response against it. So now we have lots of T cells that are capable of recognizing foreign antigens, okay? And if they bind self-antigens, they won't attack them. They won't kill our own cells, okay? So they're what we call self-tolerant, okay? So they're not going to kill our own cells. They're self-tolerant, and they can recognize foreign antigens, and they will destroy foreign cells if they encounter them. Okay, we're going to talk about APCs in a minute, but for right now, we just want to know these T cells, they're capable of recognizing foreign antigens presented to them, and they can distinguish foreign antigens from self-antigens. This is self-tolerance. Okay, they tolerate our own host cells. You know, when somebody has an autoimmune disease, that's a type of disease where the T cells, or even the B cells, but where these cells are actually attacking our own host cells, and they shouldn't be. They should be self-tolerant, but their development didn't go quite right, okay? Or their activity is not quite, it's a little bit too sensitive. Okay, so these naive T cells, at this point, these are called naive T cells because they're developed, but they've never encountered a pathogen yet. They've just been developing, and the reticular epithelial cells developed them in the thymus. So they've just been developing in the thymus. They're naive. They've never even seen a pathogen before. So now they're going to leave the thymus and they're going to colonize. They're going to go to live in lymphatic tissues all over the body. So they're going to go to the lymph nodes, the tonsils, the spleen, and the appendix, all lymphatic tissues, and reside there. And one day soon, they will see a pathogen. So we have, let's stick with our T cells. So we have 4 types of T cells. Okay, 4 types of T cells. And so here we have 4 classes. Okay, here's the 4 classes. So first we have T cells called cytotoxic T cells, or killer T cells, or I want you to know this number here. This is going to help you understand some of the other figures we have in the PowerPoint. We call them CD8 cells. This has to do with a particular protein they have on their surface. We call it CD8, that's its name. Okay, so these CD8 cells, these are the main effector cells. These are like the main killer cells of all of the T cells. They actually have the ability to destroy infected host cells. Okay, and they can become activated. When a T cell is activated, this means it has seen a foreign antigen. It's reacted to a foreign antigen. It's reacted to a pathogen. And it is now starting an immune response. That's what activation is. So our T cells, they're actually activated by infected host cells. So we're going to look at how that works coming up here. And they respond only to proteins we call MHC1 proteins. Okay, so CD8 cells, or cytotoxic T cells, they respond to MHC1 proteins. We're going to look at this coming up soon. All right, the second type of T cells are called helper T cells, or CD4 cells, CD4 T cells. And these are cells that I kind of think of them as like the admiral of all of the immune cells. They are, even though they're called helper cells, they're sort of the boss. They can secrete all kinds of chemicals that attract other T cells and B cells and even macrophage to an area where there is an infection. So they can secrete lots of chemicals that tell other white blood cells what to do. And they're essentially helping the B cells and the T cells. Okay, so they're helpers. They're helping the B cells and T cells. And these guys respond only to MHC2 protein. So we're going to look at that as well. MHC2 proteins are found only on antigen presenting cells. I know we talked about those last week. And we're going to take a look at that coming up as well, how their activation works with the antigen presenting cells. All right, and then these other two types of T cells, these aren't quite as important for you to remember, but you do need to memorize these. These are just like kind of the two main ones that I want you to know really, really well. The regulatory T cells, these are cells that have the ability to limit our immune response. So they're kind of making sure we don't have an sort of hypersensitive immune system. They're making sure we don't develop an autoimmune disease. These are cells that can suppress the activity of the other T cells. They can inhibit these cytokines or chemicals that the T cells use to promote their activity or promote each other's activity. The suppressor T cells can inhibit this cytokine secretion. So they can kind of block the T cells from being so active, and they can block the T cells from multiplying if we don't really have an infection. We need to make sure we suppress our T cells so they don't get out of control and kill our own cells, our own host cells in our body. And then finally, we have the memory T cells, and these are pretty important too. The memory T cells are descended from the other types of T cells. So here we have from cytotoxic T cells, and these are the cells that actually remember the pathogen. So these are cells that live a really long time in our body, and if we ever encounter the pathogen a second time, or even a third time, these memory T cells will start multiplying right away and develop a strong immune response so that we don't get any severe infection. We won't really even know we were invaded by that pathogen. Okay, and then we'll go on to talk about how those different kinds of T cells, the helper T cells and the cytotoxic T cells, become activated shortly. So the B cells, these are very similar to the T cells. Okay, the B cells, their site of development, they start their development in the bone marrow, and they actually finish their development in the bone marrow. So they have all of their development going on in the bone marrow. And the B cells have a selection process similar to the T cells with the negative selection test. So any B cells that react to to self antigens, right, to our own host antigens, are going to be removed. They're going to be eliminated because we don't want any B cells that will attack the cells that belong in our body. So we have a similar negative selection type of test. The B cells also, like the T cells, they have surface receptors that can recognize foreign antigens. So they can recognize foreign antigens as well. And these B cells are going to produce clones. They're going to multiply, just like the T cells did. Okay, so we're going to get lots of copies for them. And then, just like the T cells, they're going to leave their tissue. They're going to leave the bone marrow, and they're going to colonize the same lymphatic tissues and organs as the T cells. So lymph nodes, spleen, and so on. They're going to live in those organs throughout their life, waiting until they encounter a pathogen and they can initiate an immune response. Okay, so let's talk about the T cells again. Let's go back. We're going to stick with cell-mediated immunity for just a little bit here. We're going to start with the helper T cells, okay, the CD4 cells. And remember, we said CD4 cells recognize a protein called MHC2. So we're going to see, like, how does this work? How does this come into play? So the helper T cells, they have this thing going on where they can't recognize antigens on their own, okay? They can't recognize foreign antigens on their own. They can't just encounter a pathogen in the body, recognize it immediately, and kill it. That's not how they work. The way they recognize antigens is with the help of special cells called antigen presenting cells, APCs. Okay, so you definitely need to know what APCs are. These are the cells that help the T cells recognize foreign antigens. APCs are always types of macrophage. They're always phagocytic cells. So APCs, they're always, oh, my pen isn't working. They're always phagocytes. All right. Okay, they're always phagocytes. So we've got some examples over here. Dendritic cells, remember the Langerhans cells in our skin and mucous membranes, those are APCs. Macrophage are APCs. Reticular cells, not reticular epithelial cells like in the thymus, those are different. Reticular cells that live in the lymph nodes and the spleen, those fixed, those stationary macrophage we discussed, those phagocytic cells, they're APCs. And B cells, they're actually phagocytes too, they're APCs. Okay, so the way this works is that our CD4 cells, our helper T cells, they are going to use these different types of phagocytic cells to help them recognize foreign antigens. So this is what occurs. Here's our phagocytic cell. All right, it already consumed a pathogen, and let's draw the pathogen in here. Okay, so let's draw the vesicle that is holding the pathogen. And here's our pathogen. Let's make our pathogen red. Okay, here's our little pathogenic particle. Maybe it's a protein, maybe it's a whole bacterium. Okay, and this phagocytic cell is going to add some digestive enzymes to this mix that will actually destroy our pathogen. So our pathogen is going to be turned into tiny little molecular bits at this point. It's going to be totally broken down. All of the proteins inside of this pathogen will be destroyed. They'll be broken into small pieces. So now what our APC does is it takes these little display proteins. Okay, these are displaying proteins called MHC2. MHC2 stands for major histocompatibility complex 2. Okay, so there's different major histocompatibility complex proteins, different display proteins or little cell ID tags. They're like display proteins. There's different kinds. There's MHC1 also. We'll talk about that later. But MHC2 is only found in APCs. Okay, MHC2 is only found on APCs. So here, now this cell, once it's destroyed this pathogen, it's going to take some MHC2 proteins. So I'm drawing them out here like little half rectangles. Okay, so these are the MHC2 proteins. And in these MHC2 proteins, it is going to place some of these little molecular bits of this pathogen. So here's part of our pathogen. Here's another molecular piece of this pathogen. Here's another molecular piece. Here's another molecular piece. Here's one. Okay, and now when a T cell comes along, specifically a helper T cell, let's use a different color. Okay, so here's our helper T cell coming along. This helper T cell can inspect this foreign particle here. Okay, it can inspect what the macrophage ate. It can inspect what the dendritic cell ate. The dendritic cell is putting these little proteins out here to see to say hey, look what I ate today. Is this foreign? Should we start an immune response? And the T cell comes along, and this is the T helper cell. Okay, it's the H, T helper cell. And it's coming along and it's inspecting the MHC2 proteins and whatever they're holding. And the T helper cell, as soon as it binds this, okay, this helper T cell, if it recognizes that this is a foreign molecule, the helper T cell will immediately initiate an immune response. So this process of our macrophage here presenting the foreign antigen to the helper's T cell, this is called antigen presentation. And the first part where the macrophage actually broke down the bacterium and then, or the virus or the part of the virus, whatever we have here in this vesicle, this process of the macrophage actually breaking this down and then putting those little molecular pieces into MHC2, this is called antigen processing. So the macrophage is processing the foreign antigen, and then it's presenting the foreign antigen to the helper T cell or to the CD4 cell. Okay, so let's look at a diagram that shows this so you don't have to just rely on my little image there. All right, and so this is a diagram that is showing antigen processing in a helper T cell. Okay, so here we have phagocytosis going on. Whoops, let's not do that. Let's get the drawing. Okay, so here we have our phagocytosis occurring. We're taking in our little bacterium, our infectious particle, and we're going to fuse a lysosome with this vesicle containing this particle. The lysosomal enzymes will chemically digest the bacterium, breaking it down into small molecular pieces. Then little bits of that bacterium will be inserted into MHC2 proteins. And these MHC2 proteins, see how they're proteins being made at the endoplasmic reticulum? You should remember that from AMP1. In the ER, this is where proteins are made. So anyways, here's our MHC2 protein coming in. It grabs one of these foreign particles and it travels to the plasma membrane and it's holding it here. Okay, it's waiting for a helper T cell to come along and inspect it. All right, so that's our antigen processing, and this only occurs, it can only occur in APCs because antigen presenting cells are phagocytic. They're the only cells in our body that can take in foreign antigens like this and then process them, right? And then display them for inspection. Okay, so this guy is now ready to report this foreign antigen to helper T cell. All right, let's go on. Okay, that's not working. All right, there we go. So on this slide here, we have the next step, okay? This is the antigen presentation part of getting this T cell ready to go, this helper T cell activated. So what we're going to have now, here we have our APC, okay, that just processed. This is like what we saw on the last slide, but here's the APC. It's much smaller on this slide. See the MHC2 protein right there? Okay, it's holding the foreign antigen here. And we can see here is an inactive CD4 cell, a helper T cell right here. Okay, and this guy's coming along and he is going to inspect this little particle here to see, is this foreign or is this a host particle? Should we start an immune response or should we not? And remember I said we have a whole library of T cells. We have T cells that recognize all kinds of different foreign particles. So one T cell, one helper T cell might come along and it might not have the right receptor to recognize this little foreign molecule. Okay? Another T cell might come along and bind to this and it might not have the right receptor to recognize this little foreign particle here. But at some point, the correct T cell will come along and bind to this. And when it does, that is when we get recognition of this foreign antigen. Okay, so when we have the exactly right T cell, the T cell that has the proper receptor right here to recognize this particular foreign particle, then the T cell recognizes it. It's like, oh, this is the foreign particle that I'm meant to bind. Okay, and as soon as it binds, the helper T cell is going to become what we call activated. It's going to start initiating an immune response. All right, so let's see what happens once the T cell becomes activated. So here's our original helper T cell. I don't know why my pen's being funky. Okay, so here's our original helper T cell. And so as soon as it becomes activated, it's going to undergo a great deal of mitosis. So that is important to know, mitosis. All right, we're gonna just ditch the writing because it's not working. Okay, so our T cell is going to begin multiplying. Okay, lots of mitosis. And we can see here it's going to make a whole bunch of active helper T cells. And these cells, right, we're producing lots of them, okay? So we're increasing the number of helper T cells that can recognize this particular foreign particle in the body. So we're boosting up the number of T cells we have in our body that can attack this particular pathogen. Okay, so now we've got a lot of T cells, and all of these T cells start secreting cytokines. These are little chemical molecules. They're kind of like hormones, but they're not quite as long range, but they are chemical molecules that are going to stimulate T cells and B cells. They're stimulating cell-mediated immunity and antibody-mediated immunity. So these cytokines are going to attract all kinds of T cells and B cells to help get this immune response going. Additionally, this T cell is going to, while it's multiplying, produce some memory helper T cells. And these memory cells are the ones that live in our body for a very long time and can recognize this pathogen if it ever comes back again. These guys are inactive right now because we want to save them for later, okay? They'll become activated later if we get this pathogen a second time. All right, let's go on. Okay, so now let's look at, we just discussed what happens with our helper T cells and how they become activated by the antigen presenting cells. But now we want to look at our cytotoxic T cells, the CD8 cells, and we want to see how they become activated. Okay, so this is going to involve host cells that we have in our body, not antigen presenting cells, but just typical host cells. Almost any cell in our body can activate a CD8 cell. And remember, these are the cytotoxic T cells. All right, so these cytotoxic T cells, they can't recognize antigens on their own either. Just like the helper T cells, they're going to need another cell to present them with the foreign antigen. So the way that this works, now CD8 cells, these cytotoxic T cells, they don't work with the APCs, okay? They're going to work a different way. So let's draw here. This cell, this could be any host cell in our body, okay? Any host cell. It could be a liver cell, a neuron, a skin cell. It's not any cell, it's any cell with a nucleus. Whoops. Any cell with a, why is this happening? All right, any cell with a nucleus. So let's make sure we have a nucleus here. Okay, here's our nucleus. And so it could actually be an antigen presenting cell, but let's not use that example right now. Let's say this is a cell in our liver, okay? It's a hepatocyte. So if this hepatocyte, one of the things this cell has to do every single day is while it's doing its normal everyday activities, okay, there's all kinds of chemical reactions going on in here. There's all kinds of activity going on in this cell here. All right, is every day it has to take little molecular bits of whatever is found, little molecular pieces of the molecules found in the cytoplasm of this cell, and it has to place them outside on this cell in these little display proteins. These display proteins are MHC1 proteins. Okay, so it's going to take a little piece, a little molecule from its cytoplasm here and place it in MHC1, and here another little molecule in this cytoplasm and place it here, and another little molecule from the cytoplasm here and place it in MHC1 in each of these cases. Okay, so it's just taking, basically it's taking a report of whatever's going on inside the cell and placing it out here in MHC1. Now, let's say one day this liver cell becomes infected with a virus. So now little viruses are being made inside of this cell. All right, so that we have all kinds of viral activity going on inside of this cell now. So during its normal daily activities, it takes a little molecular component of whatever's going on inside the cytoplasm. Now it's going to place in here maybe one of these viral proteins. proteins. Okay, so now we get viral proteins out here in our little MHC1 display proteins. Okay, so T cells all the time will come around and check these cytotoxic T cells. So here's our cytotoxic T cell, or our CD8 cell, okay? And our cytotoxic T cell is coming around and it's gonna bind to this with its little receptor, and it's gonna check, okay? Is this protein held in MHC1, is it a host protein or is it a foreign protein? If it's a host protein, the cytotoxic T cell just unbinds and leaves. If it's a foreign protein, the cytotoxic T cell will actually kill this host cell, and then the cytotoxic T cell will become activated. So let's just use a star to show the activation. Okay, so if we have a foreign particle, if we have these red foreign proteins presented to the cytotoxic T cell, then the T cell will kill the infected host cell and it will start an immune response. It will become activated. All right. So MHC1, this protein we're talking about that displays these little foreign antigens and host antigens, just little proteins that are found in the cytoplasm of this cell. MHC1 is found on every nucleated cell in the body. Okay, so every nucleated cell in the body needs to be displaying a little report of what is going on inside of that cell. So that's why we have MHC1 on the outside of these cells. It's like a little cellular ID tag. It's a little report of what's going on inside the cell. If we have a normal host cell, a healthy host cell, then the MHC1 displays normal host antigens, okay? The T cells don't get activated then. They just inspect it, they see that it's normal, and then they move on. But if our host cell is infected, then MHC1 will display some foreign host antigens. When the T cell binds, then it can become activated and initiate an immune response. So let's take a look at this with some diagrams. All right, so here we've got our diagram, and we have here, as we can see, an infected host cell. We've got an infected host cell. So here's our little pathogen, okay? Or here's a bacterial pathogen, here's a viral pathogen, okay? So once this cell is infected, now we're starting to make the virus, or now we're starting to make, here, look, we're starting to make this pathogen inside of this cell, or there's some bacterial peptides, there's some little bacterial proteins inside of the cell. So when this cell, as part of its normal everyday activities, produces MHC1 to give a report of what's going on inside of here, it happens to grab one of these foreign proteins, one of these little foreign particles. And so now our MHC1 is going to be transported to the plasma membrane as usual to display this molecule, and it's a foreign molecule, okay, that we've got here. So when the T cell comes along, it's going to bind to this and recognize it as foreign, hopefully. So we can initiate an immune response. So let's keep going here. Okay. So now let's see our T cell binding. So here's our infected cell. Here's our infected host cell. Here it's displaying the foreign proteins in MHC1. And here comes our CD8 cell. Notice it's still inactive. Okay, it hasn't recognized this yet. So it's binding, it's inspecting it. All right, antigen presentation is going on. We're presenting the antigen. We're having binding, and this T cell happens to be the one that recognizes this little foreign particle. So we get recognition. Whoops, we get recognition. Okay. Then our cytotoxic T cell becomes activated. When we say the T cell is activated, it's going to go into this mitosis frenzy. Okay, it's going to start multiplying very, very quickly. and produce many copies of itself. So we're going to get lots of active T cells, okay, that can kill in other infected host cells. And we're going to produce lots of memory T cells that can live in our body for a long time and recognize this pathogen if it ever comes around again. Okay, one of the things I want to mention that isn't shown on this slide is that before, this is kind of like a little additional step in this process, okay? In this process right here, during antigen presentation, if the T cell recognizes that this is a foreign antigen right here, it has to sort of double check that this is really a foreign antigen. Okay, so we're going to go through a process during this T cell activation. We're going to go through a process called co-stimulation, this word right here called co-stimulation, where the T cell, before it becomes activated, it has a second protein interaction with the infected host cell. Okay, so the T cell, once it sees, okay, it looks like we've got a foreign antigen here, but I just want to double check. Okay, so the T cell binds, says, I think we've got a foreign antigen, but I just want to double check before I start a huge immune response in the body. So it goes through this process called co-stimulation. This is kind of like a verification mechanism or a double checking mechanism where the T cell actually extends another protein, another like a interaction type of protein to the infected host cell. And if the infected host cell binds that protein, then the T cell has like this double check sort of verification where now it knows, yes, this cell is definitely infected and I should initiate this huge immune response that we've got going on here. I should begin multiplying and undergo activation. Okay. So this co-stimulation step is sort of like a double checking mechanism where the T cell's kind of saying to the host cell, are you really infected? And the host cell saying, yep. And so the T cell's like, okay, let's make that immune response happen. All right, next slide. Okay, so it looks like I'm missing some words here. It's okay, I can tell you what they say. So this is the active T cell destroys the antigen-bearing cell. So as soon as it has that co-stimulation step occur and then the T cell becomes activated, we already showed that part, okay, where the T cell started undergoing mitosis, then the T cell will destroy the infected cell. So remember, this was our infected cell we just saw on the last slide. They kind of flipped the directionality of the diagram, but here we have our infected cell, and now the T cell destroys it. Okay, here's our lysed cell. This cell has undergone cytolysis. So there's a few ways this can happen. Here's two of them here. One way is that the cells can, do you remember perforins? Think back if you remember what perforins are. Perforins are used by the NK cells, the natural killer cells, to create a pore in the plasma membrane. Actually, let's not draw it like this. Let me erase that. Okay, so we've got a little pore here now in the plasma membrane of our infected host cell. And this is a giant pore that can allow basically the homeostasis of the cell to be completely disrupted, where all of the intracellular ions and components can leak out and all the extracellular ions can leak in. And so basically the cell can no longer maintain its homeostasis and it will die. The second thing the T cell can do is release a whole bunch of cytokines that stimulate this cell to undergo this process here called apoptosis. And apoptosis is it's programmed cell death, where this cell will actually initiate its own sort of molecular program, sort of like a computer program that automatically shuts down the computer. It's like a molecular program inside of the cell that automatically kills the cell. Okay, so it involves releasing some really harsh chemicals inside of the cell that destroy it. So the T cell can basically tell the cell, okay, you're compromised, you need to initiate your destruction program now, and that's called apoptosis. All right. Okay, so this is a little summary that we've got here. So I just want to outline this for you. So here we have our CD8 cells. Here we have our CD4 cells. Okay, so these are the helper T cells. All right, why is this pen so bad? These are the helper T cells, and these are the cytotoxic T cells over here. Okay, so the cytotoxic T cells are activated by class 1 MHC proteins, so MHC1 proteins. These are proteins that are found on every nucleated cell in the body. And MHC1 is indicating to the cytotoxic T cell if the cell is infected. Okay? The helper T cells, these guys don't inspect every single cell in the body. They only inspect phagocytic cells. And those phagocytic cells have the class 2 MHC proteins. Okay? Those are the proteins that hold whatever the phagocytic cells ate. So this is indicating the presence of pathogens, toxins, something foreign in the body. It's not indicating that the phagocytic cell is infected. It's indicating that the phagocytic cell found something foreign. So the helper T cell, it doesn't have any killing ability, okay? It doesn't kill the macrophage, or it doesn't kill the phagocytic cell, because the phagocytic cell isn't infected. It's just saying, hey, look what I ate today. Is this foreign? Okay. So these work a little bit differently. So I just want you to make sure you memorize what the cytotoxic T cells bind to and recognize, right? They're recognizing infected cells. What the helper T cells bind to and recognize, right? They're binding to the phagocytic cells, and they're just recognizing the presence of some pathogen in the body or some toxin in the body that the phagocytic cell consumed via phagocytosis. Okay, so once our cytotoxic T cell becomes activated, we get a whole bunch more cytotoxic T cells, we get some memory T cells, and we do get suppressor cells. Don't worry about this one right now, okay? Yeah, you just know what they are. You don't have to know that they're produced in this process. And then the helper T cells, when they become activated, they produce more helper T cells and memory cells. So basically, both of these cells, when they're activated, they produce copies of themselves that will do the same work as them, right? If the cytotoxic T cell becomes activated, we produce more cytotoxic T cells, and then we produce some memory cells. And if the helper T cell is activated, we produce more helper T cells, and we produce some memory cells. So it's very similar. It's just the way they're activated that's different. And I want you to make sure you know those details. All right, let's look at our B cells now. These are a little bit different. Let's use a color that's going to show up better. Okay, so the B cells, here we can actually see a little bit with the co-stimulation. It's okay though. All right, so the B cells, the way these work, we're going to focus on B cells now. So remember, T cells are part of our cell-mediated immunity. B cells are part of our antibody-mediated immunity. So we know the end result here, if we get activated B cells, we're going to end up with some great antibody production. Okay, so B cells have different receptors on their surface. All right, first, I just want to point something out to you. This goes with our last discussion. These B cells have class 2 MHC proteins on them. They have MHC2 on their cellular surface. So B cells, they're actually phagocytes and they're antigen presenting cells. So B cells can present to the helper T cells. Okay, and actually we're going to see that happening down here. See that presentation going on with the helper T cell. Okay, so here we have our B cell. It's inactive and it's just living in the body. It's living in the lymph node or in the tonsils or maybe in the Peyer's patch in our small intestine. And it is waiting to encounter the foreign antigen that binds specifically to these receptors right here. Okay, and remember, we're going to have a library of B cells just like T cells. So there's thousands of millions of different kinds, and they all can recognize slightly different little foreign chemical molecules. All right, so one day we are infected with a pathogen, and this pathogen has the proper molecular structure to bind to this particular B cell. And here it is, binding, okay, that one of those pathogenic proteins. All right, and so this B cell is going to become what we call sensitized. Okay, it's going to take this little molecular particle and insert it into MHC2. Okay, so it's displaying it now. It processed that foreign molecule, that foreign antigen, and now it's displaying it on MHC2. It's ready for presentation. So it waits for a T cell to come along. Here, we have a helper T cell. Remember, it's got to be a helper T cell because this is MHC2, and helper T cells are the only ones that recognize MHC2. So the helper T cell binds. Okay, here we can see the binding up close. Here's the T cell receptor. Here we've got our MHC2 protein holding our foreign antigen. There's recognition going on. There's going to be some co-stimulation. Here's this other protein right here. This is our co-stimulation factor, our co-stimulation protein. And as soon as we get binding with this co-stimulation protein, the T cell has confirmation that this is a foreign particle and that we need to initiate an immune response. So the T cell is going to become activated. Okay, so I'm going to put a little star here. Okay, the T cell becomes activated. So what does that mean? Think about what that means. When a T cell becomes activated, okay, it's undergoing mitosis. It's going into a mitosis frenzy. It's producing lots of copies of itself, other helper T cells that are activated, and it's going to produce memory helper T cells. But also in this scenario, it is going to release a whole bunch of cytokines. Here, I kind of wrote over the word, but it's going to release a whole bunch of cytokines that stimulate the B cell. And this is going to cause the B cell to become activated now. Okay. So not only does our T cell become activated, but also the B cell becomes activated. So we're going to go over here, okay, where we've got our B cell. Now, this B cell is also going to go into a frenzy of mitosis, producing many copies of itself, producing lots of activated B cells, and some memory B cells. Okay, we're going to want to have memory B cells in our body so that if we ever get this pathogen again, we can destroy it very quickly. And some of these memory B cells are going to produce what we call plasma cells. And I know plasma cells kind of reminds you of blood plasma, but don't get that confused, okay? It does not have anything to do with blood plasma. These plasma cells are large cells that are essentially antibody factories, okay? They can produce many, many antibodies per second. And see how extensive their endoplasmic reticulum is? Okay, that's even drawn in this picture. We can see how extensive that ER is because antibodies are little proteins and they're producing lots and lots of these little antibodies. And they're releasing these antibodies, and the antibodies go into our bloodstream, they travel into our tissues, and these antibodies, they're exactly like the molecular structure of this molecule over here, this initial receptor that we have here. This receptor was the one that recognized the foreign antigen. It can stick to the foreign antigen. Okay? It can stick to that little part of that virus or that bacterium. So these antibodies are now moving through our body, in our bloodstream and other fluids, and they are sticking to that foreign antigen wherever they go. Okay, and those antibodies are going to slow down that antigen. Maybe it's on the surface of our bacterium, and so they slow down that bacterium and they prevent that bacterium from continuing to harm our cells. Okay, cool. I think we had everything with that. So let's go on. We only have a few slides left. 5 slides. Okay, so let's just look briefly at the structure of the antibody. So this antibody, actually, all antibodies have a Y. See how it kind of looks like a Y-like shape to it? And the antibody is made up of 4 polypeptide chains. Does it say it on here? I guess it does say it. Okay, so see how we have this one purple chain right here, and we have another purple chain here kind of next to it, and they form the Y. Okay, so these are 2 polypeptides. We call them heavy chains, oops, because they have a very, oh boy. All right, let me get my pen. Because they have a very high molecular weight. Okay, so the heavy chains, see how they're longer polypeptides than the blue? So they're called heavy chains because they're very heavy. Their molecular weight is higher, they're bigger. And the light chains are shown in blue, they're smaller. Okay, so the antibodies have 2 heavy chains, 2 light chains, and they're made up of polypeptides. They're proteins, right? Long chains of amino acids. And let's just look at some of the features here. These are really interesting molecules. So first thing is, see how out here we have an antigen binding site. This portion right at the end here, okay? The amino acids in this region are the amino acids, and we have them on this side too, are another antigen binding site. The amino acids in this region are the ones that bind to those foreign antigens. So this is the part of the antibody that sticks to those foreign particles. We can make millions of different kinds of antibodies that can bind to all sorts of foreign particles so that we can protect ourselves against many different things, many different pathogens. All right, other things in here. At the end of our polypeptide chains here, we have a variable segment. This is the part of the polypeptide chain that is different in every antibody, right? Because every antibody has to be able to recognize a different foreign antigen. So this part of the antibody is different for each different one. Okay, it's on the other side too. Here we have constant segments. So all of the part that's not dashed, the purple and blue that has no dashes on it, those are the constant segments. Those don't change between different antibodies. Okay, they're just always the same amino acid sequence. Other things in here, we have some disulfide bonds. These are sulfur-sulfur bonds where we have 2 sulfurs covalently bound to one another. These are very strong bonds that help give the antibody its shape and hold these chains together. And then we also have here some complement binding sites. Do you remember complement proteins? Okay, the complement proteins, we talked about them. They're part of the second line of defense. They have the ability to bind to these complement binding sites. So here's our little complement protein. And remember, we said the complement protein can hook this antibody up to a red blood cell. Okay, so we could draw a big red blood, very, very big here. Okay, much bigger than this antibody. And this red blood cell could then tow this antibody and whatever stuck to it out here at the antigen binding sites to a part of the body where macrophage can destroy the foreign antigens. So this complement binding site is used for immune clearance. You can look back in the PowerPoint or review your notes to see all about immune clearance again. That's that molecular towing mechanism that involves the red blood cells and the complement proteins. This basically clears the pathogen. These complement binding sites help clear the pathogen out of the blood. All right, and then here, look at this. We have a site for macrophages to bind. So any pathogen that's coated in antibodies, right? Let's say these antibodies are sticking all over to the surface of our pathogen here. Let's say there's our pathogen out here, okay? Macrophage will come along, and as soon as they bind to this, they know to consume it and whatever it's connected to. Okay, so these antibodies, once they're bind to pathogens, they also like the complement proteins, they kind of butter up those pathogens, signaling to the macrophage with these binding sites right here, like, hey, this is something pathogenic. It should be removed from the body. So that's what they're doing with these binding sites. They're helping those macrophages destroy the pathogen. All right. So let's just look at some of the different types of antibodies. This is pretty simple. This is kind of a memorization thing. Okay, so antibodies, we call them immunoglobulins. Does it say this word back here? Oh, no, I don't see it. Okay, I'm going to write it out very slowly down here. Okay, they're called immunoglobulins. Yeah, I don't see it on this slide. Okay. Oh no, this isn't going to work. Let's see, do I have it? Okay, here's what I can do. I'll just make a little text box. Okay, so we have, let's see. We have what we call immunoglobulins. Okay, great. Okay, so these antibodies, we call them immunoglobulins, and that's how we get this IG little naming system here. See how it says IG, IG, IG. Okay, and then why do we have another text box here? We don't need this. It's okay. Let's leave it. Okay, so these immunoglobulins, these are, it's the same, it means the same thing as antibody. Okay, these immunoglobulins, they're immune proteins. They're little proteins in our blood that help our immune cells destroy pathogens. And so here, that's where we get this name. And so we name them with different letters. So this type of antibody that we see in this first, over here on the left, this is called IgG. Okay, and so we're just using the letter G to be able to recognize this one, to name this one. Here, this one has a slightly different structure. It's called IgE. Here, this one has a different structure. We call this type of antibody IgD. Okay, and there's different kinds of each one. There's different IgG molecules, all that recognize different foreign antigens. There's different IgE molecules, all that recognize foreign antigens. There's different IgD antibodies, all that recognize foreign antigens. So let's look at some of these different kinds. There's two others on the next slide as well. So IgG, this is the largest class of antibodies. We have in our body, this is the most abundant type of antibody we have. They're 80% of all antibodies are IgG. These are the ones that are responsible for resistance against lots of viruses and bacteria. And these are the ones that cross through the placenta. So these are the ones, and also they're, oh, okay, never mind. Okay, so these are the ones that can cross over the placenta, and they can confer passive immunity to the fetus during development. So these are very protective to the developing fetus. Let's see. Oh yeah, let's not worry about that part. Okay, so these are the antibodies that can cross the placenta during development, and they are very protective for the fetus. Okay, so the mother, basically, she has these in her blood, and these can cross through the barrier in the placenta. They can cross from the mother's bloodstream into the fetal bloodstream and hopefully protect the fetus from having any type of disease develop during its development. IgE, we have next over here. IgE antibodies are the antibodies that are on the surfaces of basophils and mast cells. Okay, so basophils and mast cells, remember, those are the ones that release histamine and also heparin. And these cells, when they encounter an antigen that binds to this type of antibody, when they encounter an antigen that binds here to IgE, those basophils, so if our basophil, this would be our basophil, okay, here's IgE attached to it. Here's IgE attached to it. Now, if we have some foreign molecules in our body that come and bind to these antibodies, the basophil or mast cell will release histamine to start inflammation in this area. So this is really important for kind of initiating the immune response. Next, we have IgD. And so this is what I want you to remember. I want you to remember kind of the main characteristics, the main function. So IgG, this is the one that if we have a viral or bacterial infection, it increases in number a lot. This is the most abundant type of antibody we have. IgE is on the basophils and mast cells and is important for initiating immune response by causing those cells to release histamine. So do you guys get the idea? I want you to kind of memorize the main features of these. IgD is found on the surfaces of B cells. So on that on one of those last couple slides where we're looking at the B cells with those little receptor molecules on their surface, those are antibodies, okay? And so here's our B cell, right, with its IgD on the surface. And if that B cell encounters a foreign antigen that can bind to this, that will sensitize the B cell, and then the B cell can present that antigen to a T cell, hopefully initiating an immune response. So these are the ones found on the surface of B cells. IgD. Here we have two more. So IgM is when we have encountered an antigen, okay? When we encountered a foreign antigen the first time, we start producing lots and lots of this antibody called IgM. And see how it's sort of like an antibody complex. See how we have multiple antibody structures in here, and they're all linked together in this big pentagonal shape. Okay. And so let's see. During our immune response, we're going to look at this coming up here, but during our immune response, first we produce a lot of this, and then our cells start producing more IgG. So IgG and IgM are really important in our immune response. Okay, these the anti-A antibodies and the anti-B antibodies, remember those we talked about in lab, these have those antibodies have this structure as well, okay, anti-A and anti-B antibodies. All right. And then our last type of antibody is called our last class of antibody is called IgA. And IgA is found primarily in secretions from our body. Okay, so it's found in mucus, in tears, in saliva, and in semen. Okay, there we go. And so if you can think about it, in our tears, right, that kind of they're present in our eye, they kind of coat our eye, and they're very protective to us. This antibody is found in those tears, and basically we want antibodies in our tears because if any pathogens come onto the surface of our eye, we would like those pathogens to be neutralized as soon as they encounter the surface of our eye. So hopefully these IgA antibodies that we see here, see how they have a very unique structure? These two antibodies bind in the middle with the secretory piece that allows them to be released into our fluids. Hopefully, this antibody will bind those pathogens as soon as they encounter that fluid, as soon as they encounter the tears. And if that pathogen tries to migrate into our body, it won't even matter because the antibody will already have been bound to it and be protecting us from infection by that pathogen. So I just want you to remember these are found in our different secretions that we have listed here. Okay, so those are our 5 classes of antibodies, and they all have slightly different functions. Okay, I want to talk about how the antibodies are how the antibodies work, how they attack pathogens. So we said earlier, antibodies don't actually kill anything like cytotoxic T cells do. Okay, they're just proteins, they can't kill anything. So this is what they do. They work in 4 different ways that we can see on this slide. First thing they can do is actually neutralize the pathogen. So they can actually bind to the pathogenic region of the antigen. So maybe our bacterium has some proteins on its surface that it specifically uses to infect our cells. Or maybe the virus has a specific protein portion that it uses to inject its DNA into our cells. So what the antibodies can do is actually bind to that infectious part of the virus, preventing it, like blocking it from actually infecting us, from actually injecting the DNA into our cells. That way, that virus can never really truly harm us. Okay. Another way antibodies work is the second mechanism here called complement fixation, where the antibody will, once it binds to the foreign antigen, it changes its shape and it allows that complement binding site to be exposed so that complement proteins can grab onto it. And once the complement proteins bind and or are activated, then we get all of these different effects down here. If you remember from the other day, we get inflammation, we get phagocytosis, we get cytolysis, and we can get immune clearance as well. So the antibodies have the ability to help activate those complement proteins and then clear the pathogen from our body. And then down here we have agglutination. The antibodies can bind to the enemy proteins or the enemy antigens and immobilize them. So maybe they just bind all over the surface of the bacterium, slowing it down so much in the body that it can't really get anywhere, and then eventually a macrophage will come along and consume it. And then last mechanism of antibody attack is called precipitation, where the antibodies actually bind to the foreign antigens, creating complexes where they're kind of stuck all over those foreign antigens in groups. And then they kind of become insoluble, so they don't really move around much in our fluids anymore. And then eventually, in antigen antibody complexes, those are very easily eventually phagocytized by eosinophils. So eosinophils are very good at consuming these, and they can do that easily. Okay, so our last slide here, we have a slide on our primary and secondary immune responses. So first thing up here, our primary immune response is the immune reaction we have when we have our first exposure to an antigen. So it's brought about by our first exposure to an antigen. So this is the very first time we ever see this pathogen. Okay. In this process, in this primary immune response, our B cells are going to multiply, turn into and produce plasma cells. Then the plasma cells will make antibodies, and we'll get some memory B cells as well. Okay, so hopefully when we get to our secondary immune response, right, maybe our second time or our re-exposure to the same antigen, we already have a lot of these cells. We already have plasma cells in our body, right? We already have memory B cells in our body. So we can hopefully remove this pathogen from our body before we ever get sick, before we ever really have any symptoms. So let's just look here at these graphs, okay, for our primary immune response and our secondary immune response. So this is on the y-axis, our serum antibody titer. It's basically telling us the concentration of antibodies in our blood, okay? Basically telling us how much of the antibody we have in our blood. And on the x-axis, we have days from first exposure to the antigen, or here, days from re-exposure to the same antigen. So in the primary immune response, see how it takes like 3 or so days Like we're first exposed, okay? And then it takes a few days before we even start producing any antibody right here. Okay, and see how we're producing IgM. That's that really big one with those 5 branches. Okay, and those antibodies level, the level of that antibody rises for several days. Okay, and in that time, we also start producing a lot of this other antibody, a lot of IgG. Okay, and so we get our peak levels of antibody right around day 15 or so. So this is when we have our greatest level of antibody production, around day 15 after exposure. So at this time, the pathogen should no longer be causing us symptoms. Now let's look at our secondary immune response. In this graph, we can see that we have, even from day 0, so when we first encountered the pathogen, we already have so many memory cells in our body that that pathogen is going to be detected on the first day of exposure, of re-exposure, I should say, okay? In the beginning, we didn't have any memory cells, and we had very few copies of that particular clone of B cells. So we didn't detect our pathogen right away. It took us some time to detect the pathogen, and then it took us time to start producing antibodies as well. But now we already have memory cells. So not only do we detect it really fast, but we start producing antibodies very, very quickly. Those memory cells can be activated really fast, and the plasma cells can start producing antibodies really fast. The plasma cells already exist. So even now by day 3, okay, we already have a higher level of antibody than we have ever had here in our primary immune response. So this is why when we encounter the pathogen the second time, and look how much IgG we have, okay, by day 4 or 5, okay, very high levels. The antibody binds to and prevents the pathogen from causing us symptoms. Our T cells and our macrophage clean it up, and we never even know we were re-exposed. Okay, let's see. So that's all written here. Now the last thing is that our memory does not last as long in humoral immunity as in cellular immunity. And the reason for this is because the cells, the T cells, like the memory cells, the T cells, and even the memory B cells as well, they can live a very long time. Okay, so that's a very long-lasting type of immunity involving the cells. Humoral immunity, and humoral is referring to just the antibodies. The antibodies do not last a long time in the body because they are just proteins. So these little antibodies, while they can be present in our blood in very high concentrations, they don't have a very long life in our blood. They don't last for years and years. They can last for weeks, but they don't last for years and years. Now, this doesn't mean that years later we can't produce a very high concentration of them very fast, just like we did right here, okay? But those individual antibodies are broken down in a matter of weeks, and then they leave our blood. So the cells last much longer than antibodies do. Cells typically last much longer than proteins. Okay, so that is the end of this lecture, and I hope you enjoyed it. If you have any questions, please feel free to email me. You guys will, so my Santa Fe class, you guys will be having a quiz on this on Monday, and my College of Central Florida class, we will be having a quiz on the lymphatic and immune system on Thursday of this week. Okay, I hope you guys have a great weekend, and I will see you soon.